We're going to travel back, all the way back to 2008, where Obama said, yes, we can, where Aviv Regev was the Overton Prize winner at ISMB, where the world was taken over by jellyfish. I'm not sure if many of you remember. And when this fresh-faced Curtis looked a little bit like this, and clearly he's aged very, very well. The stresses of the job haven't got to you. So this is back in 2008. Curtis interviewed for a faculty position at Harvard School of Public Health. And for those of you who don't know much about the faculty interview process, I'll just set the scene a little bit. So it varies between institute and countries, but normally it involves some kind of formal presentation to the institute, which Curtis has already given in this case. Maybe some one-on-ones with a few different PIs within the institute, and then a style of talk which is commonly known as a chalk talk, which is no formal slides, maybe a whiteboard, and it's very much about your research plan and where you're going. So we're going to pick up halfway through that chalk talk procedure. So Curtis has already gone through his research plan, and I hope most of you have had a chance to read that plan. It was sent out as part of the materials. Check your emails and bring it up on the laptop if you haven't got that already. And so he's sat down, he's in the interview, and he's going to be quizzed about how he's going to run his research group and how he's going to fit into the faculty in this position. So interview panel, take it away.
So thank you, Curtis. My questions have to deal with how you're going to start up your lab and what kind of personnel you envision hiring as first off, and what your approach to supervision of your lab might be.
Okay, so something that I think is particularly important in this particular context, which as background was the biostatistics department at the School of Public Health. Something that I think is important in this particular context is the degree to which I want to incorporate wet lab component into my research. This is something that I found particularly valuable during my own PhD and postdoctoral work. And it's a little unique in this context. So I know we had talked before the interview a little bit about what a wet lab here might look like. And it's something that I don't want to necessarily start up right off the bat, but it's something that I would have planned to start over the first few years of my faculty work. That means that immediately over the first 1 or 2 years, I want to focus on getting in somebody like a postdoctoral fellow to be able to help with research as quickly as possible to start the publication pipeline. Somebody like a software developer to be able to support our computational work, and something that I'm particularly interested in, as well as making sure to push out tools and data to be publicly available. And then eventually, probably something like year 2 or 3, having somebody come in at a senior level to really help steer the opening of a wet lab space, since it's not something I want to do with my own hands, but it's something that I wanted to have as part of a component of my research. Did that answer some
And what might your approach to supervision of your lab and students, how might that look like?
So I enjoy interacting as much as possible with individual lab members, with individual projects. I plan on having something like weekly one-on-one meetings and then a weekly group meeting with the lab. My experience during my PhD, for example, we had a rotating presentation schedule. That's something I'd like to carry on here. And this is something that's evolved over the years. But thinking back to 2008, it's nice to be able to interact informally with individual lab members as much as possible as well, such that folks are able to stop by with a question or with a manuscript or problem that they're working on, in addition to the formal one-on-ones and formal weekly full lab meeting schedule.
So like an open door kind of policy.
All right. So I read your proposal for future research. You propose to work on functional conservation, pathway prediction, regulatory codes, metagenomics, microflora, tissue specificity, temporal specificity, and data formats. I'm not convinced you have a clear focus to your research. Actually, I have to say I'm very much concerned that you're overreaching and stretching yourself thin. So how will you develop a clearly focused research agenda?
So it's an interesting reading of the research plan. I can help to focus in on the areas of interest there. The three areas that I'm particularly interested in for the short term at this point are split between human-focused work and microbially focused work, which is very much like what I did during my PhD. During my PhD, I did a collection of yeast functional genetics studies that we construed as microbial, and a collection of human molecular functional studies that were human systems biology and disease focused. And those are the two areas that I'd like to continue forward. On the human side, the big opportunity that I would see next is taking some of the first steps that we've done in multi-omic data integration in human molecular studies and making those more specific. So what we've done is, or what I've done over the past year or two on the human side, has included ways of building molecular networks from multiple different data types. What I'd like to do as a next step is make those molecular networks as tissue specific or pathway specific or disease specific as possible.
So this is a continuation of your PhD work. So your
in
supervisor in Princeton is working in the same area. How will you differentiate your research from hers?
So on the microbial side, an area that I'm personally interested in is microbial community studies. This is not directly related to yeast work, and certainly not something that, to the best of my knowledge, is going on at Princeton, but is something I see as a huge opportunity. It's an area that really needs a lot of computational work, and it hasn't been tackled by in 2008 especially, hasn't been tackled by the bioinformatics community at all. And it's an area that I just find personally very interesting. There's a lot that we don't know about how microbial community molecular biology works in. So that's an area that I think particularly differentiates me from Olga Trinskaya, my advisor, from some of the other work going on in the field.
All right, Curtis, I'd like to ask you about teaching. So an important part of your role here is going to be educating and mentoring graduate students. And so I'd like to know something that you thought that your advisor, Olga, was particularly inspiring about, that you want to emulate, and something you didn't like about the way she taught or mentored graduate students that you want to do differently.
Interesting. That was definitely not a question I got to. So no one here is going to report back to Olga.
It's on video.
So something that I particularly enjoyed about Olga's teaching style, both taking her classes and then helping out later as a TA, was her degree of enthusiasm about the entire subject area of bioinformatics. Olga did a great job of communicating both biology and computational material in a very enthusiastic and excited way, and really drew people into the field that way, I mean, myself included. I think maybe something that I want to focus on more than Olga did during her teaching is certain areas of technical depth, especially from the computer science side. That's something that I have in my background that wasn't necessarily part of Olga's background, that I miss teaching. I used to do more pure computer science teaching, introductory programming. It was something that I really enjoyed doing, that I haven't had an opportunity to recently at Princeton, that would be, I think, fun to incorporate into education in the biostatistics department going forward.
Okay.
So as the lab becomes productive, obviously some of the output is papers, and authorship is always a contentious point, both within the lab group, you know, who gets to be first author, et cetera, et cetera. and also within the bioinformatics sphere, because you're often collaborating with a group that has the data set, and bioinformatics they're collaborating with you to do the bioinformatics analyses. And what is, if you could describe your approach to authorship, both within your lab group and the larger bioinformatics context?
I think within the lab group, something that I would envision for organizing projects in general, and including authorship, is to make sure that everyone, students and postdocs particularly, come in with a main project, or a potential main project, I'll say. Especially in the case of postdocs, I think it's important that they have something that is there, that if they become interested in it, and if it goes well over the first 6 months or 9 months, becomes their main thing. And thus that's their main project, and they're the first author on that thing. What, again, especially for postdocs and to a degree students, what I'd expect to be the case over time is that either that works and that's the thing that they're excited about and the thing that they do and they're first author, or they get excited about something else or get involved in collaborative projects, at which point there's the possibility to diversify out around that initial thing. So that provides at least a potential way for each student or postdoc to have a thing that they own that they're first author on. For collaborative projects, that's a case where, especially in a community like Boston, where there's just a lot of everything going on all at once, one thing that's good about that is that the community is used to collaboration. What I would expect to be the case for bioinformatics is to make sure to discuss openly and negotiate up front who has what responsibilities and what that might mean about authorship, assuming that things go according to plan. I think it's important to keep tabs on who's actually doing what, and thus who's actually eligible for what authorship over time, but making sure, just like for individuals, that there's at least one plausible plan that's been discussed up front is something that I would think would be important.
So you said you like teaching computer science, but this is a biostatistics department, and I'm very worried about your lack of formal training in statistics.
Me too.
Your graduate school doesn't even have a statistics department. So how are you engaging, do you plan to engage with the biostatistics faculty here at Harvard School of Public Health, and how will you teach our students statistics?
So something that I've been interested in for this position in particular is the degree to which the biostatistics department has been interested in growing out computational biology. And I'm personally curious to see how that goes forward over the next few years. I know from talking to folks like John Quackenbush that part of this involves more collaboration with the Harvard Computer Science Department, more collaboration with the MIT computational biology program, more collaboration with quantitative biology groups like systems biology. At this point, what it looks like is that, projecting back to 2008 again, at this point what it looks like is that there's some incipient work in the biostatistics department, specifically in computer science. We teach an introductory computing class that I can imagine being developed in at least two ways, one of which would be into more computer science for the life sciences and for biostatistics, and another of which would be into computational biology. So I can see growing the Comp Bio program in at least those two ways, to incorporate computer science both for our biostatistics students and for the surrounding health professionals, masters in public health students, molecular and bench biologists, all of whom will need a degree of computing for modern biology, and in building out more methods-based bioinformatics courses that I think would be more specific to biostatistics. So if either or both of those two programs are actually what's of interest in the department over the next few years, I can see contributing to one or both of them.
Yeah, now this is a question that nobody at Harvard would ever ask, but I'm in Colorado where we care about these things. So I'm curious, our department really has, I think, a positive spirit and a community where we work together and try and support each other. I want to know how you're going to contribute to this culture that is this joint thing that we create. How are you going to make a positive contribution to the environment that's not just advancing your research interests or your students?
Do you want me to answer for the Harvard department or the Colorado department? It'd be very, it would be.
They're kind of different, but why don't you, well, you give me both.
I'd say to help fill in some context for the audience too, one of the big differences I'd characterize is between a soft money environment at Harvard, where there's a big push towards specifically getting grants to sustain each lab, and a hard money environment where there's more sort of centralized infrastructure and departmental education program and things like that. So is that the kind of organizational difference?
Well, no, I'm at the med school. We're all soft money too.
That's true. Okay.
But no, just other context. So in my department, we really want all of our junior faculty members to get tenure. And in Harvard, they mostly look down the hall and say, okay, there's 12 of you. We want the best one of you to get tenure and the rest of you to find jobs somewhere else.
A little bit, yeah. So I'll try to hit somewhere in between as far as integrating into the department. Something that I think is particularly important in the medical area at Harvard, which I alluded to in the previous answer, is the degree to which the biostatistics department provides a large part of the quantitative skills and quantitative methods for the whole life sciences community. So something that I think will be an important opportunity for me as a computational biologist in this community is bridging some of the really quantitative theory and methods oriented areas in biostatistics with some of the applications in bench biology or in translational biology in the clinic that happen right next door. So I think sometimes it's a little hard to go all the way from somebody doing theoretical statistics to somebody working in a cancer lab. But I've seen a couple cases from talking with current faculty where computational scientists provide a nice bridge, and I'd like to help occupy that part of the community as well over the next few years as somebody who understands the quantitative methods, but is particularly interested in making concrete tools or concrete analyses that might help drive something. And personally, my interest is more on the molecular side, reaching towards the translational end a little bit. So did that somewhere in there answer the question?
It's okay. What do you think about the state of quantitative education for biologists and physicians? And would you be interested in playing some role in trying to define what quantitative education those folks ought to have?
Something, and that relates to the previous question as well, something I'd be particularly interested in doing as far as teaching, in addition to, say, a core computer science course like introductory programming, would be, I'll call it an applied bioinformatics course for bench biologists. There exists, and did exist in 2008, a health sciences focused bioinformatics course that is at a very high level. What I'd like to hit is something that's more molecularly focused, not a methods course for biostatistics students, but something that would allow students and postdocs doing molecular biology work to understand a little bit of computing and a lot of bioinformatics methods applications, and build a course around that for that audience, because that's a big audience in the Longwood medical area.
So I noticed from your CV that you just recently completed your PhD and that you're postdocing in the same lab. And the position is, a tenure track position, and most other applicants have several postdocs coming into this. I'm guessing. I don't know what.
Statistics is a little different.
I don't have no idea. Okay. Can you walk right out of a PhD into a tenure track position? It seems. Tell me about what distinguishes you from other candidates who might have a deeper experience.
There's a couple of things. One is the biostats department tends to hire people without postdocs.
Okay.
And I would editorialize that I actually, in retrospect, I'm not a huge fan of what I did. I think doing that was a bad idea.
Mm-hmm. It flagged on your CV and I don't know the Harvard context, but or the departmental context.
And there's two different things going on. One is that honestly, biostatistics departments often hire without a postdoc, which is what it is. Two is that I honestly don't think that was the best career move for me. Like I think I would have done better as junior faculty had I done a longer, more normal postdoc in a different location. So I got that additional breadth of experience. To answer as I would have at the time, I've had I've enjoyed my PhD research, and I think been exposed to a pretty good diversity of application areas within the particular set of computational methods development that I've done for biological data integration and molecular network analysis. At this point, I'm excited to work on some of the more I'd say PI typical tasks of grant writing or personnel management in order to help expand that research program. I know there's a limit to how much I can do myself as one person, even during another postdoc, such that I'm curious to see how much more we can do in that area or in the area of microbial community studies, as per the earlier answer, by building out a group around that research program.
What will be the title of your first grant application?
That's cheating, that's an easy one. I think my first grant application, for safety's sake, will build around my main PhD research area and branch out fairly conservatively from that, focusing on what I talked about earlier, the tissue and cell type specificity of some of my molecular work, specifically working on computational methods that could be used both in that area and in microbial community studies, since cell type specific in microbial communities actually means organism specific, which is a big research need in computational microbiology more generally. So I don't know if I can provide a title, but the topic and the aims of that first grant will be building methods that can be applied for tissue and cell type specificity of human data integration, as well as organism specificity of microbial data integration.
All right, last question. So tell me where else you've applied. Imagine you got offers from everywhere. What would it take for you to come here?
I'm trying to remember where I was at the time in the interview process. To approximate. At the time I was interviewing at Harvard, I think it was late in the game, and so I had interviewed at a couple places internationally. I spoke with labs in Aberdeen, Scotland, and in the Netherlands that would be more human biology focused, and I had spoken with the computer science department at Cornell that would have been more computationally oriented and as I was saying to the panel earlier, focused on agricultural bioinformatics, which is not particularly an area of work in Boston. I think at this point, something that I'm excited about in Boston especially is the translational opportunities, the research community, and the applied clinical research here is really a differentiator, not just from the other places that I've talked to, but from most research communities. There's a lot going on, and a lot specifically in translational biology. At this point, the deciding factors would include some practical considerations, and this is again kind of speaking from I think where I was at the time. Some practical considerations on the relocation side, and in making sure that the computational areas that I'm interested in will integrate well into both the quantitative side of the biostatistics department and this applied perspective that I'm interested in translationally, but is not something I have a lot of experience with coming from Princeton, which doesn't have a medical school, doesn't have a big translational biology component. So I think what I'll be doing in the couple months ahead is weighing those options, both some things, like I said, on the practical side, and then scientifically choosing between a focus on translational life sciences versus more sort of basic biology in a computational environment versus still human-focused biology, but in a very different academic environment in some of the international locations.
Okay, great. I'll just interject. So, firstly, please thank you very much to Curtis and the panel. That was a bit of an experiment, and I think it worked quite well. Just before we wrap
You're
up this
rehired.
session, yeah. So, my first question is from the audience: from what you saw, would you hire Curtis or not? Put your hands up if you'd hire him. I think that's a majority. So if we're going on a majority vote, you've got the job. Well done. Just for the panel, was there anything in his answers that raised a flag or had any concerns?
Oh, sure. So,
I'm very curious about this.
well, so let's just take that last one. Okay, so I was looking for something much more specific. You know, I need the money to do this. I want to work with this collaborator. Now, Harvard, nobody at Harvard would ever ask the question, what would it take to get you to come here? Because they just think by offering you,
I'd like to
you want
bunch
to
in.
talk.
I mean, I was actually in the team that wanted that. Well,
yeah.
we sat around. Should we hire Curtis? Okay, and then the tick box you sign off, but then you all have a chat, right? And the majority of the team wanted him, and the worry is that he wouldn't come.
No, well, maybe you do occasionally worry
We
that
took him out
somebody.
to dinner, and it was your wife, right? We took your wife, and we knew that she liked to prepare food, so we took her to a really nice restaurant and found out all the clubs that prepare food, and everybody talked about food for the whole evening. So we tried and convinced them to come, so it was actually a very positive environment. But I wasn't on the review committee, but the word that came out of the review committee was, we want him, now we have to make sure he signs the letter. Right? So, and I never told you that, Curtis, but I thought it'd be
Well, that's some of what I was. I wasn't sure quite how much to go on to the the
Personal stuff.
side,
Sure.
but yeah, that's kind of what I was alluding to.
we
is
figured
that-
it was personal.
that was part of the decision.
And to the panel, what were you most impressed with in terms of the
So I
answers?
also think you ducked the question about what you didn't like about Olga's teaching.
And that was intentional.
Quick question. So you completely dodged the statistics question, right?
Mhm.
No, was that a negative from the panel? I mean, or did he? I mean, he basically said, yeah, I like computer science. So
And that
That's
was
true,
not
right?
an intentional dodge.
No.
That's like that is both is and was, I think, my perspective on it. And Florian, I should say, interviewed for the same position the year before I did, and also got the offer.
I also got an offer.
Also got the offer.
The way I answered it, because I know the other faculty there at this department, there's Shirley Liu and there's John Quackenbush. I just asked them, how does John Quackenbush teach statistics? And they laughed. And that's it. So that was the end of that discussion.
And that's why it was not an intentional dodge.
Yeah.
It's just the biostatistics department there has over the years legitimately become interested in expanding into computational biology. And the price in terms of the department of doing that is getting people whose expertise is computational biology and not statistics. So like I don't want to pretend to be a statistician because I'm not one. And they have those and they do good work, but it's not my work.
So in terms of do's and don'ts, so is it possible to come up with a couple of do's and don'ts based on this interview?
Well, at least in the statistics question, I would say fess up to your problems, right? So you laughed and said, yeah, me too. I don't know how I'm going to teach statistics. And so if you really have a problem area, either they're not going to want to hire you because you have the problem, or you're going to address it head on and say, look, I'm not going to teach statistics here. I'm not a statistician. You guys are the statisticians. This is what I want to teach, or this is how I want to work here. And so don't try and sugarcoat things that are really a problem. Be honest about it, and talk about what you're going to do instead. But you were absolutely right not to try and pretend to be a statistician.
A do for me is always think through the whole aspect of the job, not just your research aspect, but that's why I was asking questions about authorship, lab composition, supervision, because if you're not thinking through those aspects of your lab, you can't go in day one and start something. You've overlooked something. I think they're just as important.
So one last question.
A question for the panel, and then for the organizers. Was this
planned in advance to have the different panel roles, as in the objective managerial person, the competitive research person, and then like the general, it's like, it's all good, I'll ask you something about the social aspect, that's teaching. That's actually a pretty nice panel.
It was not planned.
No.
Which is very different.
Okay, well, I think that wraps up that session. I really hope you enjoyed it. I did. I learnt a lot from it. That was great. So we're going to change up things a little bit. Oh, sorry, one last thank you to the panel. And please